Thank you for being here and giving us an opportunity to introduce you to Silicon Valley Barcode of Life. We're grateful to have become aware of DNA barcoding early in our lives and early in its history. We see possibilities for DNA barcoding to become an increasingly important tool for cataloging our diverse biological heritage and for motivating people worldwide to protect it and secure its many benefits for us and for future generations. We hope that after learning about what we're doing with DNA barcoding, you'll join us in acting to realize its potential to further common good. To begin, we have a few questions for you. Do you have an estimate of how rates of biodiversity loss today compare to average extinction rates during the past 50 million years? What is the International Barcode of Life? And how does biodiversity in the San Francisco Bay Area compare with that in other parts of the United States? We'll fill you in on these things and more in the next few minutes as we reply to 5 basic questions about Silicon Valley Barcode of Life. Those are, what is SVBOL? Why does it exist? What do we do? Who are we? And what can you do? What is Silicon Valley Barcode of Life? We're people with Silicon Valley ties working with nature to change hearts and minds. We're learning about why biodiversity is important to human well-being and acting to protect it. SVBOL is DNA barcoding. We collect samples of organisms present here in the Silicon Valley and its surroundings, send them to a lab where scientists analyze small segments of DNA to generate identifying barcodes, and upload findings to a publicly accessible database that can be used to document biodiversity here and how it is changing. In the process, we learn and we teach others about biodiversity. We perceive that increasing people's understanding of the value of biodiversity is means to further common good. SVBOL is one of many initiatives that contribute to the International Barcode of Life, a global consortium dedicated to protecting biodiversity. Why does Silicon Valley Barcode of Life exist? SVBOL is a response to the 6th Great Extinction, a global catastrophe created by humans and now underway. It's rooted in the International Barcode of Life, the organization I mentioned just a minute ago, and it reflects my sister's and my personal stories. Humans are part of and are sustained by a web of diverse life. With our increasing numbers and ever more powerful technologies, we're disrupting ecosystems worldwide in ways and at speeds beyond other species' capacities to adapt. Today, we're driving them to extinction at rates 100 to 1,000 times greater than those which prevailed for the past 50 million years. The last extinction of this scale occurred when a meteorite impact with as much energy as billions of nuclear bombs ended the reign of the dinosaurs. We've answered our first question. Humans are extinguishing species at rates 100 to 1,000 times greater than those that prevailed for the past 50 million years. Most of us are careful to avoid losing even a small part of a single day's work. The information contained in Earth's diverse life is the work of 3.5 billion years of evolution. Conservation biologists estimate that we'll lose a quarter to a half of biodiversity in the 6th Great Extinction now underway. We're burning our library of life. Silicon Valley was once called Valley of the Heart's Delight. Now it's Valley of the Superfund site. Even over our short lifetimes, my sister and I have observed landscape degradation and loss of biodiversity here where we live and in the Sierra where we vacation most summers. Native plants are disappearing, invasive species are spreading, insect pests are multiplying, beneficial insect populations are collapsing, bird numbers and variety are in decline. SVBOL has its roots in the International Barcode of Life. Here's how SVBOL began. Once upon a time, a Canadian biologist named Paul Hebert traveled to the tropical rainforests of New Guinea to study butterflies and moths. New Guinea covers only 0.5% of global land area, yet it's home to an estimated 5 to 10% of global biodiversity. Paul collected insects hundreds of times more rapidly than he was able to classify them. As he recollects, they wrestled me to the ground. I'd collected enough species to keep me occupied for the rest of my life. He stopped collecting, packed his belongings, and moved his research to the Canadian Arctic. Canada, the second largest country in the world, is 7% of global land area and contains an estimated 1% of global biodiversity. There, Paul was able to classify as quickly as he collected. Paul was less than satisfied with this so-called solution. 20 years later, he was still pondering how to classify his tropical collection. Inspired by grocery store barcodes, Paul wondered if he might apply advances in DNA sequencing to create a DNA barcode that was able to distinguish species. In partnership with others, he identified relatively small genetic regions for animals, plants, bacteria, and fungi in which organisms of a single species showed little variation, and those of different species showed substantially more. Taxonomy is still in the analog era. Since ancient times, it's been based upon morphology. It's slow, expensive handwork, limited by available expertise. Although biodiversity has been studied for thousands of years, more than 90% remains to be classified. DNA barcoding is means to bring taxonomy into the digital era. Barcoding is a way to assemble a library of identified species more quickly, reliably, and inexpensively than is possible using only traditional taxonomic methods. People with minimal training can directly and effectively participate. Engaging diverse segments of the population in documenting biodiversity is a demonstrated way to increase people's understanding of its value and motivate us to protect it. With barcoding, we reveal hidden species. There's a surprising level of unseen biological diversity, literally in front of our eyes. For example, researchers using DNA barcodes discovered that a well-known skipper butterfly, first identified in 1775, is actually 10 distinct species that feed on 10 different plants. The urban environment can also be remarkably diverse. In New York City community gardens, students use DNA barcodes to catalog 54 species of bees and 24 species of butterflies. To take advantage of the many benefits of barcoding, Paul Hebert and collaborators from around the world form the International Barcode of Life. SVBOL goals are to DNA barcode by 2035 all multicellular species on Earth, to create a library of life with DNA extracts from all multicellular species, and to globally monitor changes in biodiversity. We call this the SVBOL Moonshot. SVBOL exists to cooperate in this effort. Now we've answered a second question. The International Barcode of Life is a global biodiversity conservation initiative. It includes national, local, like Silicon Valley Barcode of Life, and taxa-specific groups collaborating to DNA barcode all multicellular species, store DNA samples extracted from them to form a DNA library of life, and globally monitor changes in biodiversity. People often ask us, why are you doing this? For as long as we can remember, we've been exploring the natural world. From the time we were very young, we've worked on habitat stewardship projects. Here's 4-year-old Hillary planting native bunch grasses, and here I am leading volunteers watering recently planted native oaks on local open space. When we were 10, we met Dan Jansen and Winnie Hallwachs, conservation biologists from the University of Pennsylvania. Since 1985, Dan and Winnie have led in creating Área de Conservación Guanacaste, the largest protected area in Costa Rica. They've been DNA barcoding flora and fauna of ACG since 2003. Almost 5 years ago, we visited them in Costa Rica and learned firsthand about their research. When we returned home, Brad Zlotnick, the friend who'd introduced us to Dan and Winnie, described to us San Diego Barcode of Life, which he'd founded. We wondered, can we do something like that here? We think Silicon Valley is a great place for a DNA barcoding initiative. It's recognized worldwide for technological innovation, so what we do is visible and may lead others. It's full of people with expertise applicable to DNA barcoding. It's also home to many who want to further common good and have the means to do so. Perhaps most importantly, we have a wealth of biodiversity to conserve and protect. San Francisco Bay Area is one of the top 6 U.S. hotspots where biodiversity and rare species are concentrated. With Silicon Valley Barcode of Life, we may well discover yet-to-be-named species in our own backyards. Now we've answered the third question we asked you at the beginning of this presentation. Next, let's look at what we at Silicon Valley Barcode of Life do. We bring people together to learn about the importance of biodiversity, threats to it, and ways each of us can act to protect it. We partner with public and private property owners and managers to monitor selected sites, collect and prepare specimens, sequence DNA to generate barcodes, identify associated organisms, and upload data to Barcode of Life data systems, freely available online worldwide. BOLD Systems already contains over 6 million barcode records. We can use these to identify much of what we collect, and we can add to them with our specimens. Our first fieldwork event was a DNA barcoding BioBlitz at Hidden Villa. About 100 people attended, and together we collected nearly 400 mostly unique arthropod specimens. We're currently organizing photos and data for each specimen and preparing to send the samples in for sequencing. Here we see an overview of the DNA barcoding process: specimen collection, tissue sampling, DNA processing and sequencing, and managing data. Now we'll take a closer look at some of these steps. The first step is to collect a specimen, preserve it, and note its geographical location and local environment. Tent-like malaise traps are commonly used to collect flying insects. It is easy to gather leaf samples from plants by hand. Next, we sample tissue. An insect leg or a small leaf disc are suitable sources of DNA for barcoding. 2 organelles, mitochondria and chloroplasts, are key for barcoding because they contain short, highly variable regions of the genome which are similar among individuals of the same species and different from one species to another. Interestingly, the genes used for barcoding are involved in the key reactions of life, storing energy in carbohydrates through photosynthesis and releasing it to form ATP during respiration. With hundreds to thousands of copies per cell, chloroplast and mitochondrial DNA sequences are readily amplified by polymerase chain reaction, even from very small or degraded specimens. A typical animal cell will have on the order of 1,000 to 2,000 mitochondria. A typical photosynthetic leaf cell from a plant, like wheat, contains about 100 chloroplasts per cell. For animals, the region of DNA that is sequenced to create a DNA barcode is about 700 base pairs from the mitochondrial gene CO1. CO1 is involved in the electron transport phase of respiration. For plants, scientists have had more difficulty identifying an effective barcode region. Barcoding leaders currently recommend RBCL and MATK, 2 gene regions in the chloroplast. RBCL is the most abundant protein on Earth and catalyzes the first step of carbon fixation by photosynthesis. We extract DNA from the tissue sample and amplify the barcode portion of RBCL or CO1 with PCR. This slide shows the denaturing of DNA into single strands, the attachment of primers, and the addition of new nucleotides to form double strands. Each cycle of PCR entails these steps and doubles the number of copies of DNA. Then we read the DNA barcode with Sanger or SMRT sequencing technology. Finally, we upload complete sequences to Barcode of Life data systems, search for matches with species already represented, taxonomically examine voucher specimens for species with barcodes different from those already present in BOLD, and place these in a phylogenetic tree with near relatives. BOLD now holds millions of specimens with barcodes representing hundreds of thousands of species. Who is creating Silicon Valley Barcode of Life? We're people who work, live, or have other ties to the Silicon Valley. We're nature lovers, landowners and business people, artists and designers, life and computer scientists, government officials and employees, teachers and students, parents and children, and members of civic groups. We're advised by eminent conservation biologists and other leaders of pioneering barcoding projects in the U.S. and abroad. We're guided by local experts. All of these companies and other organizations contributed goods and services. What can you do to create SVBOL? You can contribute expertise in everything from identifying insects to graphic design. You may be able to provide or arrange access to land to collect specimens. You can collect and prepare samples. You may be able to sequence DNA or arrange for local sequencing, and you can give money or material. Finally, you can contribute to SVBOL by learning about biodiversity as you are today, by advocating conservation of biological resources of our planet, and by communicating about how all of us depend on biodiversity and can protect it. Thank you very much for devoting some of your life to learning from this presentation today.